Hello everyone, I am Dr. Sirajul Faizi. I welcome you in my YouTube channel. Today in this video, we are going to learn about protein. So, let us start learning about protein, especially amino acid. So, I will cover the amino acid sequence and basic concept of protein in this video. You know friends, protein is a large molecule. It is made up of chains of amino acid and it is found in every cell in the body, are involved in most of the body's functions and life process. The sequence of amino acid is determined by DNA. Greek words proteus means holding the first place. Swedish chemist Berzelius suggested the name, and most abundant molecule of living system and constitutes about 50% of cellular dry weight. My friend, first protein to be sequenced, insulin, Friedrich Sanger won Nobel Prize for this achievement in 1958. So, we will focus the structural difference between carbohydrate, lipids and protein. So, you know the micronutrients, carbohydrate, lipids and proteins. Chains of glucose, fatty acid, amino acids. Example, glucose units, triglycerides and amino acids. Now, what is amino acid? Amino acid containing carboxylic acid. You can see the carbon, which contains C-OH groups, NH2 groups, alkyl groups and hydrogen, and has chiral. And the another form, COO minus, NH3 plus, and the almost same structure. So, amino acid has a central carbon atom to which 4 different group of atoms are attached. An amino group and a carboxylic groups, a hydrogen atom, a radical or R groups which determine its properties and function in protein. Now, the acidic and basic properties of NH2 and COOH group make the amino acid molecules amphoteric. Primary amino acid or alpha type, the amino acid or amino group is usually attached with the alpha carbon and placed on left side with respect to the spatial configuration. So, L-alanine and D-alanine, you can see. All amino acids except glycine show optical isomerism and optical activity, as they have at least one asymmetric carbon. Nomenclature by the first 3 letters of their names, serine, exception, tryptophan, isoleucine, asparagine, and glutamine. Now friend, classification of amino acid based on the structure of side chain are and their reaction in solution. Acidic monoamino acid, dicarboxylic acid, asparagine, glutamine. Basic diamino group, diamino and monocarboxylic acid, lysine, arginine, histidine. Neutral monoamino, monocarboxylic acid, rest 15 amino acids. Based on polarity of side chain, polar amino acid have hydrophilic side chain with without charge. Acidic amino acid negative charge, basic amino acid positive charge. Other amino acid are of without charge. Non-polar amino acid have hydrophobic side chain with no charge. Rest 8 amino acids. Now, nutritional classification of amino acid. Essential amino acid 8 in numbers, semi-essential 2 in numbers, needed for children, and non-essential amino acid rest 10 amino acid. So, might lead 20 amino acid, essential amino acid and non-essential amino acid. So, within the body, tyrosine and cysteine are synthesized from the amino acid phenylalanine and methionine respectively. So, tyrosine and cysteine may become essential amino acid if the dietary supply of phenyl and methionine is reduced or absent. Metabolic classification of amino acid based on their catabolite or catabolic fat. Gluconic or glycogenic amino acid having carbon skeleton possessing metabolic potential to produce glucose, glycine, alanine, glutamine, valine. Ketogenic amino acid having ketone bodies, and both gluconic and ketogenic amino acid. So 20 amino acid found in protein. Now, properties of amino acid: colorless, crystalline, water soluble, high melting point, stereoisomerism, DL isomers, optical isomers, and optical activity. Chemical classifications or properties act as ampholytes because C-OH group acidic, donating proton, and NH2 group act as base, accepting proton. Acts as buffer and have definite isoelectronic pH at which molecules carry no net charge. From salt with acid or base by reacting with ammonia and C-OH groups respectively. Form ester with alcohol, forms carboamino compounds with carbon dioxide, gives color reactions with 9-hydrine, undergo acetylations and amphidations reaction due to NH2 group. Functions of amino acid: act as building block of peptides, polypeptides and protein. Arbitrary peptides, polypeptide and protein are made up of 2 to 10 amino acids, 10 to 100 amino acid and greater than amino acid respectively. Support glucogenesis is fasting and starvation. Participate in synthesis of specialized products. Example: neurotransmitters, hormones, purines, pyrimidines, heme, etc. Source of S-cysteine methionine methyl groups made in water. Definitions: macromolecular polymers of amino acid linked together by peptide bond. Minimum molecular weight 5,000 to 8,000. Number of amino acid greater than 100. Elementary composition of protein: carbon, oxygen, nitrogen, hydrogen, sulfur in different percentages. Covalent bond formed by joining the C-OH group of one amino acid and NH2 group of another amino acid with removal of one molecule of water. Amino acid 1, 2 and peptide bond. You can see here how peptide bond formed by removal of water. Covalent bond sharing of electron pair, shows partial double bond character, rigid, planar and do not rotate, not broken by denaturing agent, broken by proteolytic enzymes. So, functional classification of protein: types of protein, function example: structural protein, support framework of cell, catalytic proteins, catalysis, transport protein, hormonal proteins, gene regulatory proteins, protecting proteins, receptor protein and contractile protein. Now friends, simple protein contains only amino acid without any non-protein substance. Fibrous protein, scleroprotein, fiber-like animals origin possessing high tensile strength, water insoluble, highly resistant to prototype enzymes, chief component of tendon, ligaments, cartilage, hair, nails, example collagen, elastin, keratin, etc. Globular protein. Oval, spherical in shape, water soluble, digestible. Example: albumin, globulin, histone. Conjugated protein composed of simple protein along with non-protein prosthetic substance. Example: nucleoprotein, histones, nucleic acid. Lipoproteins, VLDL, HDL. Lipids: glycoproteins, mucin, carbohydrates. Chromoproteins, hemoglobin, cytochrome, heme. Metalloproteins, ceruloplasmin and carbonic anhydrase. Derived proteins: denatured, degraded proteins of simple conjugated proteins. Proteins, metaproteins, proteoses, peptones, peptides, amino acids. Protein and metaprotein, primary derived protein, has no little change in peptide bond. Rest are secondary derivative proteins produced by progressive hydrolytic cleavage of peptide bond. Nutritional classifications of protein: complete protein contains all amino acid in essential amino acid in proportion required for body. Example: egg, albumins, meat. Incomplete protein lack one more essential amino acid. Example: gelatin. And partially incomplete proteins. Now friend, best source of protein. Proteins are abundant in dairy foods, meats, poultry, meat alternatives such as dried beans, peanuts, butter, nuts, and so on. A well-balanced diet can meet daily protein needs. Most people consume adequate protein from their diet, do not need protein supplements. Now, properties of proteins: isoelectric point of protein, colloidal proteins, protein denaturations, protein precipitation, protein sedimentation, protein hydrolysis, color reactions, UV light absorption. Now friend, isoelectric point. PL is the pH of an aqueous solutions of an amino acid protein at which the molecules on average have no net charge. So structure of protein: 4 levels of organizations: amino acid residue, secondary structure alpha helix, tertiary structure of polypeptide chain, quaternary structure a simple subunits. So any alternation in the structure or sequencing change the shape and function of protein. Now primary structure means what? Exact sequence of amino acids held together by peptide bond in peptide chain. It is determined genetically, and this is the terms determine secondary, tertiary, and quaternary structure. Example: insulin. You see the primary structure. Secondary structure: helical or pleated sheet-like conformation produced by definite periodic folding, twisting, or coiling of primary structure. Hydrogen bond is present here. Example: fibrous protein, collagen, elastin, keratin. Alpha helix: most common and stable conformation of a PP chain, spiral structure, rigid right-handed, stabilized by hydrogen bond. Distance between amino acid are 1.5 degree. So you can see primary structure, secondary structure, alpha helix. Beta pleated sheet PP chains are almost fully extended. Distance between amino acids are 3.5 degree. Stabilized by hydrogen. Major structural motif of protein. You can see beta pleated sheets. Adjacent strand in the sheet can run in the same direction with regard to the amino and carboxylic end of the PP chain, or in opposite directions. You can see anti-parallel top view side. Parallel top view side. Now friends, three-dimensional globular form of protein produced by further folding, twisting of secondary structure, itself with the hydrophobic side chain, buried interior, and hydrophobic groups exposed inside. You can see the tertiary structure. This is myoglobin. Now quaternary structure. You can see the quaternary structures. Now factor affecting the protein structures: hydrogen bonding, electrostatic interaction, hydrophobic interaction, van der Waals forces, and disulfide bonds. You can see